all. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Today, I have a vaccine that I wanted to discuss with you. It is quite an interesting and fascinating vaccine. So if we think about it for a second, what are the common issues or objections about the vaccine? So let us start to think about that in a second. One is just generally that somebody is anti-vaxxer, and they say we just do not believe in it, or it is harmful, or whatever is their reasoning. Just in general, they're not going to take it. Fine. So that is a group of people that we cannot sit here and try to convince. That's one. Then another objection is vaccines have not been tested for long enough. They have not been built for long enough. They have not been trialed in animals and so on. So that is another objection, which I think is going to be applicable for all vaccines. So if somebody doesn't want to use a vaccine because of the shorter duration, then all vaccines are going to fall in that category for some more time. Then there is a common objection that the vaccine may have some things in it, for example, RNA or DNA, that is going to go into our cells and kind of mutate our cells, which is not the case, but that is an objection. Then we have a very common objection that once the vaccine is given, let's say here in the deltoid, and the nanoparticle or the adenovirus is picked up by the tissue here, including the muscle cells and the mast cells and the fibroblast and other tissue cells, those cells are going to be eventually killed by the natural killer cells or cytotoxic T cells. So that is a destruction of the cell. So that is another objection. Then there is an objection that vaccine RNA, when it gets into our cells, it would just continue to make more and more spike proteins, and we do not know when it would stop. Although I have done this discussion many times, even an individual topic, but that objection stays. Some of these are just myths. Some of these are just objections, but still these objections are there. Then there are objections that the adenovirus itself, we do not want a chimpanzee adenovirus in us. Although many of the vaccines other than SARS-CoV-2 as well use chimpanzee adenovirus, that adenovirus is actually better for us compared to human adenovirus, because human adenovirus, so for example, if my adenovirus are taken and then used, most of the people have their bodies already conditioned, their immune systems already ready to kill those adenoviruses because they live in us. And so as soon as you use them in a vaccine and you inject that into somebody, their body is just immediately going to kill them and destroy the vaccine. It would not be effective. But there is a discussion about adenovirus. Now within adenovirus, then there is another objection which I receive very commonly, is that the adenovirus has a DNA. That DNA would enter our nucleus, which is our DNA. Over there, it is going to make messenger RNA and spike proteins outside in the cytoplasm. But in this process, because the DNA has gone into our DNA, that is somehow going to be an issue. Then there is another objection about the adjuvants, that some vaccines have adjuvants in them. And those adjuvants can cause reactions. Then nowadays, we know that there are reactions to the vaccine because of polyethylene glycols, and maybe that is an issue as well. Excuse me. So how about if we see a new vaccine today which removes majority of these concerns? And of course, I forgot to mention the fetal tissue usage issue. For example, AstraZeneca, or for example, other vaccines like Johnson & Johnson yesterday, we talked about. These are manufactured in the copies or the clones of the cell coming from aborted fetus. And that is a area which some religious groups, not only Christians, but other religions as well, have objection to. Then there is a gray, excuse me, gray area of the objection where a vaccine may be designed in using a fetal tissue clone cell, but is not manufactured in it. And that example is, for example, Moderna. So all of these issues, imagine a vaccine that can bypass majority of those. And that vaccine is Novavax's vaccine. So I'm going to talk about that vaccine today. So let me very quickly go over the same list once again and just explain that why does it not, why these objections are not applicable to it, or majority of the objections. One objection that the vaccine is developed in a very short time, that is applicable to all of these. And so Novavax would have that, that objection applicable as well. Other objection that it is not tested on animals or it is not tested for long enough, that also is applicable to Novavax's vaccine as well. Then the question of messenger RNA coming into our body, that is not true for Novavax. The question of adenovirus or a DNA coming into our body, arriving in our body, bringing the vaccine in, that is not true about the Novavax. Objection of DNA accidentally entering our nucleus and doing something to it, that is not applicable for Novavax. Objection about the RNA hijacking somehow or making somehow proteins forever, spike proteins, by hijacking our ribosomes, that is not true for Novavax at all. So the Novavax, the fetal tissue issue is not true for the Novavax at all. So Novavax has a vaccine technology that bypasses and sidesteps all of these issues. The adjuvant issue may be something that is to be looked at because they also use a nanoparticle-like technology. In their nanoparticle, do they use polyethylene glycol or not? I do not know yet. I'm researching. So I'll do some more discussions, but majority of this issue is actually gone. Then one more issue, that the cells that are displaying the spike protein, for example, muscle cells or fibroblast, and these would be destroyed. That is also not true for Novavax, because it is not picked up by the muscle cells. So muscle cells are not going to be destroyed by this vaccine or due to the presence of this vaccine. So majority of the issues are actually sidestepped by it. So it is a fascinating technology. Let's look into it. All right, so this is Drbeen.com. If you wanted to buy, just to have access to these lectures or to support, you can actually go here and check the plans. So that is one. Then here, it's a fascinating thing today that we are going to be discussing. I love this one. So for this discussion, for the Novavax technology to understand it better, one, we need to understand the spike protein genetics. And we know them from our previous discussions. I have the references here, just so that they are available. We have seen this reference again and again, and this is again about the spike protein of the SARS-CoV-2. This is another reference here, which is about so this whole bar that you are seeing here is the genetic code of the spike SARS-CoV-2. And then here, this little dark green part is the genetic segment of the SARS-CoV-2 genes that are responsible to make spike protein. We are talking about these today. Then this is the Novavax phase 3 trial, December 28 news. And they are starting their phase 3 with 30,000 volunteers, placebo-based. This is Novavax's own site with their phase 3 trial information. And they are recruiting at this time. So if you are eligible and if you wanted to take part in it, you can. This is their technology itself. And I had touched upon this a few days ago as well. Today, we'll actually look into it in a more deeper way. This technology is called recombinant nanoparticle vaccine. Recombinant is an important word here, and then nanoparticle. Then here, this is their phase 1 and 2 clinical trial link. I have the link in the description. These are the cells that they have used, and they are not fetal tissue cells. They're not even human cells at all. They have taken the cell from butterflies or moth. And so we're going to talk about that. So with these links in front of us, let us now look at the vaccine itself. Beautiful technology. I loved it. So the vaccine is called NVX-CoV-2 373. So NVX standing for the Novavax, CoV-2, and number 373. It is a recombinant nanoparticle technology. So it's not just the messenger RNA-based nanoparticle, it is a recombinant-based nanoparticle. So how does it work? So here, they take the SARS-CoV-2's gene, then they take the spike protein gene area, which starts from the 21,563 kilo base pairs to 25,384. So this is the area, this part is the part of the SARS-CoV-2 genome that is responsible to make spike protein. So they take that part of the genome. Then what they do is this. So here is a moth, and I should have made actually a butterfly. I missed it. I should have made a pretty butterfly over here. So a moth or a butterfly, their larva here, and they take the cells from there. So the question of the cell acting as a manufacturing site is still there. So if you said, I don't want my vaccine to be built in a cell, then we have a problem here because there is a cell involved. And majority of the drugs have cells involved. But if you said, I don't want human fetal, aborted fetal tissue cell copy used as a manufacturing unit, then that is not involved. These cells are called SFN BV cells. And I'll explain what does that mean. SFN are for these cells that are coming out of the moth or butterfly. These are fall armyworm moth cells. So once you take those cells, here is the BV part. So the cell culture name or the cell line name is SF9BV. So what does BV stand for here? So these are the baculoviruses. These viruses actually infect the cell itself, the moth cell or the butterfly cells. And ignore this thing over here. I had made a copy to use it again, but I forgot to take it off. So here is the Bculovirus. I think you can actually see this area of the Bculovirus. It has spikes on it as well. Most of the viruses have spikes on them to bind with our cells. So in this case, this Bculovirus is not going to bind with our cell. It is going to actually bind with the moth larva cells. The structure is very simple. It has this head area or an apical area where it has the spikes to bind with the cell, and then it has little DNA area where it has its DNA, just like adenovirus has its DNA. And this red area around that is the capsid protein or the cover of the DNA. What they do is they take this DNA of this virus and they change it to add the spike protein DNA to it. And so, of course, you can ask me this question that why the heck we're talking about the spike protein DNA? Why not spike protein RNA? So yes, from the SARS-CoV-2, we get the spike protein RNA, but we convert that RNA into a complementary DNA and then a DNA, and then put that into this DNA. This is the recombinant part. We use recombinant technologies. Recombinant simply means to change a DNA. Now, this is not a DNA changed in us. This is not a DNA changed in our cell. This is a DNA changed in a virus. And that virus would never end up in us. For example, when we talk about adenovirus-based technologies like AstraZeneca, or Johnson & Johnson, or CanSino, these adenovirus-based technologies have a virus that has a DNA which is altered with the spike protein or contains the spike protein. Here we have a virus, Bculovirus, that has a DNA which is altered to add spike protein genome to it. Good. Now they take this infected virus or modified virus. Are you with me so far? So what have they done? They took the moth cells. First of all, from the SARS-CoV-2, they extracted the RNA part that makes a spike protein. That's one. Then they took cells from butterfly larvae and they infected those cells with Bculovirus. But these Bculovirus are already prepared by adding in them that spike protein genome in them. So this is a modified Bculovirus. This Bculovirus, if you see here, is now inserted into the SF9 cells, the moth or butterfly cells, not us, those cells. Do you know that this is very similar to the adenovirus vaccine that was being injected in us? So instead of the vaccine being given to us, imagine the vaccine is now being given to butterfly cells. So what happens when we talk about AstraZeneca or Johnson & Johnson? When the vaccine is given to us, our cells make spike proteins. Here, the virus or the vaccine, in a way, is given to the butterfly cells. So what are these cells going to make? They're going to make spike proteins, correct? So look at this. The virus binds with the butterfly cell, then it gets in there, it releases its DNA. That DNA enters the nucleus of the butterfly cell. I would keep saying butterfly cell, so you don't take this that somehow our cells are being affected. This all is happening outside. Now, this DNA piece here is from the virus, and this little red piece on it is the spike protein genome from SARS-CoV-2. When this spike protein genome becomes transcribed into messenger RNA, comes out again in the moth cells, butterfly cells, SF9 cells. This mRNA is going to go to their ribosome, and their ribosome is going to make the spike protein. Correct? So this is like our muscle cell was going to do this in case of AstraZeneca or in case of Johnson & Johnson or CanSino. Here, we have a hired cell, the moth larva cell. So the spike proteins are made there. Now what do we do? What is our function in this? When these spike proteins are formed, our now job is to extract the spike proteins from them. So in AstraZeneca and in Johnson & Johnson, they would give us the vaccine which will go into our cells and ask our cells to make spike proteins. Here, we have given the job of making spike proteins to an external cell. And that is not a fetal tissue cell. And now that cell is making spike proteins. Guess what is the next step? They pick up those spike proteins that are made by these cells. We steal those spike proteins from them. Just like when the bees make honey, we take their honey, correct? Just like that, the infected cells, moth cells, are going to make spike proteins, and we're going to take the spike proteins. Then we convert those spike proteins. This is another beautiful part of their technology. We convert those spike protein into a particle that has the same size like SARS-CoV-2. So they join these spike proteins on a lipid particle, and the size of the particle is very similar to SARS-CoV-2. So now imagine, you may have seen sometimes hunters, when they go for, let's say, hunting birds, they have decoy ducks, and they put those decoy ducks in the lakes, and then they hide and they're trying to hunt. So here we have a decoy SARS-CoV-2. It is not a real SARS-CoV-2. It is actually just a lipid nanoparticle. It has nothing in it. But on its surface, it has many spike proteins attached. So they call this immunogenic nanoparticle. Beautiful. They just, instead of calling it normal nanoparticle, which would contain inside it a messenger RNA that needs to get into our cell and then cause the production of spike proteins, here the nanoparticle itself has spike proteins on it. Interesting. So now the first question I want to answer here: can this nanoparticle bind to our ACE2 receptors and then get into our cells? No. It can bind to our ACE2 receptors, but the TMPRSS2 and all other functionality with the spike protein is not available. The enzymes are not available. So this thing can do nothing, even if it enters a cell. So that is the beauty of this. So this is a decoy SARS-CoV-2-like particle. It has no brain in it. It has no RNA in it. It has no matrix protein in it. It has no N proteins in it. It has no capsids. It has nothing. Just a particle with the spikes on it. Now on the right side here in this diagram, I have made the actual, not the actual, a compared SARS-CoV-2 as well, which has all of its other proteins in it plus the RNA. And here is the decoy nanoparticle, which they call immunogenic nanoparticle. So let me actually show you this much on their site. So why these things excite me are that there are researchers, there are intelligent people sitting out there who think about these things, and then they come up with these technologies and make these things and advance us. So here, if you see, what they're saying is that, hey, we will take the genetic material of interest, spike protein. We will then infect a Bculovirus with it. So here is a virus, their depiction of virus. Then we will take that virus. So did I say infect? They would change the virus genome, Bculovirus genome. Then they would take that virus and infect the moth cells or SF9 cells, which are insect cells. Then those insect cells will now make the genetic protein that we wanted to, or the immunogenic protein, for example, spike protein here. Then they would generate particles that look like the original pathogen with the spike proteins on its surface, or whatever antigen we want on wherever it should be. That becomes the technology. And this video is beautiful as well. Last time I had said that I have the video in the description, and somebody said, where is the description? So under the video, let's say if you're on YouTube, if you just expand it, under the YouTube, there is a video, there is a description. In the description, there are links to these areas. Okay, so back here. So hopefully this much is clear. Now what happens is we bottle up these spike proteins, and now we are ready to inject them into a human being. So it doesn't have adenovirus in it. The lipid nanoparticle, I still have to see the composition, that does it have polyethylene glycol in it or not. It seems like from their video, it seems like it does not, but I have to confirm yet. It doesn't have any messenger RNA in it. It doesn't have any DNA in it. It does not have any such things. It doesn't go into the RNA, into the cell and give it the RNA and make spike proteins that may continue to be made forever and all. So all those myths or objections or concerns are not applicable here. So now we have the spike proteins, and we know this diagram very well. Once we inject that spike proteins, let's say in a muscle, now muscle cell is not going to pick them up. It is the immune system that is going to pick them up. Why? What is the difference of this nanoparticle compared to AstraZeneca's nanoparticle, for example? The difference is that AstraZeneca's nanoparticle, or CanSino's nanoparticle, or Johnson & Johnson's nanoparticle does not have any immunogenicity on its surface. It has the RNA in it, and it would like the nanoparticle. I spoke the wrong names. They are all adenovirus based. Moderna and Pfizer. Their nanoparticle has the messenger RNA in them. And that nanoparticle has to get into a cell, release the messenger RNA there to make the spike proteins. Here, we're talking about a particle that already has the spike proteins on it. So our immune system will directly interact with it. Immune system would directly become interactive. So we don't need to get it into a muscle cell or a fibroblast or a mast cell or some other cell. It just immune system cells, dendritic cell, macrophages, natural killer cells, neutrophils, they are all going to come around this little particle and they're going to say, my God, what the heck is this thing? What are we talking about? What the heck is this? And they would start working with it. So here, let's say we have a dendritic cell. The dendritic cell is going to bind with the spike proteins, just like if it was a SARS-CoV-2, and it is going to pick it up and phagocytose it and eat it up. Now, it is not a SARS-CoV-2, so there is no worry about the infection. And now the dendritic cell is going to present that on the surface of on its surface, right? These cells are called antigen presenting cells. So on the MHC2 complex of the dendritic cell or macrophage or B cell, this antigen will be presented. When the antigen is presented, then we know that the naive T cells, the CD4 helper cells, naive or zero cells or null cells, they would connect here. And then in the presence of the correct interleukin or absence of interleukin 12, for example, they would take one route or the other. So we know this, that if interleukin 4 is present or interleukin 12 is absent, then the naive T cell, zero cell, will become T helper 2. And T helper 2 cell, which is also a CD4 helper cell, will then release interleukin 4 and 5 and 10 and others. And one big part of that is that it is going to convert a B cell into a plasma cell. And plasma cell is nothing but a pregnant B cell that is making antibodies. And these antibodies are the ones that are going to be used against the actual infection. And many of these B cells would become memory cells. They would go to sleep. And when the actual virus comes in, then they would attack it. Similarly, if interleukin 12 is present, then the naive T cell can become CD4 T helper 1 cell. And then it would release interleukin 12 that would cause cytotoxic T cell, which is called CD8 cell, to become active, which would then actively start killing any cell that would become infected by the virus. So this is the technology. You can tell how excited I am, because in this one, again, there are going to be folks who are going to say, regardless, we don't want to have a vaccine, we don't like a vaccine and all that, and totally cool, I understand that. But all of those extra objections are removed by this. And so they are in phase 3 trials right now. I would do some more discussion later on that what was the data from phase 1 and 2. But today I wanted to talk about Novavax's technology itself. So let's just see some questions and then we would stop. So let's see. Ryan Cole says, still concerned about antibody-dependent enhancement reactions as individuals are exposed to new spike mutations. Ryan, let me tell you something. So this is an area where many people are concerned, and this concern is not very valid. And let me give you an example. If this was a correct, valid thing that was happening in practice, and this is not a theoretical phenomenon, then people who have become infected with actual SARS-CoV-2 will continue to develop ADE all the time, because when the reinfection would occur, which occurs all the time, right? So if I have become infected and have recovered, now my immune system is ready. And if the virus arrives in my body again, immune system is going to start working. In that process, the antibodies are going to be present. And the new clone that comes in, it's not going to be 100% the same as before. So ADE does not occur. Otherwise, we would have seen those millions and millions of people who have become infected and are still in hospitals and are working and going around. They would continue to develop ADE all the time. we would have no choice but to get stuck in the homes and never get out. So ADE is not a very important mechanism for the vaccine concern. It is a theoretical mechanism. Okay, so let's see. So TD, this is a objection for which I cannot probably respond. If we say that testing on 40,000 people is not sufficient, or testing on 30,000 people is not sufficient, or now administration to 10 million people is not sufficient, then there is no answer to that. The way I think about all of this is, there used to be a time that, I give this example before as well, when from here, Cupertino to San Francisco, if you're going to go by a horse, you might reach there in days. And now you can reach there in half an hour. And so if we say that, why is this vehicle faster now? That is just because we have advanced technologies. Similarly, for the vaccine production nowadays, we are just fast because we have so many advanced technologies and the whole world is focused on it. All of our time is collectively looking at this. So I am not too worried about that part, but still, some people have made this comment that, hey, we are just going to wait. We are going to see others. So that may be a choice as well. So Jim Mattox, Jim, how are you doing? So would this may be a better vaccine if you're on cancer treatment that harm your B cells? Correct. So if your B cells are generally on the reduced side, any of the vaccines would work fine. But this vaccine would directly work with the immune system instead of using our naive, our other cells as well. So that is the benefit of it. It doesn't need to bring in mRNA or DNA or adenovirus and do that. But the eventual outcome for the B cell is the same. Those vaccines want the B cell to become active. This one also wants the B cell to be active. Samina says, please talk about Oxford vaccine. I have done that discussion. So if you look at my videos and just search for Oxford vaccine, that was one of the earliest vaccines I discussed. John Snyder says, just feed the new spikes to the bugs and make new ones. Yes. So that is the recombinant part. You just keep making copies of the RNA. So, excuse me. So you can't give the spikes to the bugs because their immune system is going to kill the cells that would contain the spike. But you can continue to give this virus to the bugs and they would keep making more spikes, and then you just keep taking the spikes. TD says, can you interview a vaccine genius for us? Absolutely. Do you have someone in your mind? Carolyn RN says, question, of all the different vaccines that we have discussed since last summer, or is being developed, which do you like the best? This one. This one has no messenger RNA. It has no adenovirus in it. It has no inactivated virus in it. It has directly spike proteins. And these are made into insect larvae, and insect have a much more reproductive power. They have a lot of numbers. So if we took one larva and used that cell to make their copies, hopefully that is okay. So from a technology and from the mechanism of action, I love this one. And then it doesn't need to bring in RNA, doesn't need to bring in DNA, doesn't need to have our cells do the production. It just brings in a bunch of spike proteins. That's what I love about it. Joe says, hi, Dr. Bean, I feel uncomfortable getting an mRNA vaccine. I see about 3% of those who have been vaccinated with Pfizer and Moderna have had bad reactions. Should I wait for the J&J vaccine? So I think that the problem with the Moderna and Pfizer has been the polyethylene glycol that is present in the nanoparticle. Correct to wait for J&J, because even this one, Novavax, may have, they say we use nanoparticle. What I do not know is exactly what is the usage, because, so short answer first, that yes, wait for J&J. And the longer answer is, if you see here in their video, their nanoparticle.
Traditional vaccine of antibodies targeted toward these key defenses, distinct seeing nanoparticles.
See, their nanoparticle structure is like this. So it is not the whole big lump of the nanoparticle with the spike proteins from its surface. To me, it seems like these are the spike proteins, and then there may be small, tiny holding part in it. Because of this little diagram that they have, and I'm hoping that a company that develops this would have a better and more accurate depiction compared to mine. So if this is a structure, then these spike proteins are present, and nanoparticle is really negligible here. So they may also not have the bad reactions. So good question, Joe. Simple, Steve says, a company is developing nanobodies as opposed to antibodies that will bind to spike proteins and prevent attachment to S2 receptors. Yeah, that is a very workable solution as well. Morris says, isn't the spike protein of Novavax an mRNA from a specific SARS-CoV-2 strain, and that may not generate immune response that can react to the new mutant strain now? So I have talked about this a number of times. So far, there are lots of rumors. Every day there is a new news that this strain or the variant may not have the right, the vaccine may not work correctly. Or I had done this discussion that monoclonal antibodies may fail, some RT-PCRs may fail. So far, out of thousands and thousands of mutations, there has been no mutation that would render a polyclonal antibody cocktail like Regeneron or a vaccine to fail. And now we have started seeing the data from the vaccine companies as well to say, yes, it will not fail. So I will not become too worried about this, that this actual spike protein that is in it may be different. And what they can do is, let's say right now, 90% prevalent spike protein is type, let's call it A. They can use that RNA and have that built. And then let's say a year later, there is a different prevalence of a different type, for example, UK strain. Then they can very easily start building with that. But even UK strain, or we shouldn't call it strain, variant, even UK variant is very susceptible to previous immune responses that have been generated. So far, there is no variant that has escaped the previous immune responses. So wrong IP address says, do all the non-attached spikes on the particle get antibodies attached? Does this spread through your whole body then, not injected into your muscle? So better chance of generating IgA antibody? No, so they are not given in blood. So they cannot go to the whole body. So they would be given in the muscle, and in that area, they will be immediately picked up by the immune system cells. Now, the first part of your question, that will these non-attached spike proteins, although they are attached to the lipid nanoparticle, get antibodies attached to them? So yet we are suspecting that the people don't have the antibodies. So because of that, it is the macrophages or the dendritic cell or the B cell that are going to pick them up. Now, let's say somebody had been infected previously. Then yes, as soon as their immune system is going to see that, oh man, the spike proteins are here, they would attack with the antibodies, and antibodies are just going to go bind with the spike proteins. That will make the spike proteins yummy for macrophages who would eat them up and digest them and clear them, which is a phenomena which people say that, hey, this would cause antibody dependent enhancement. But that is a normal mechanism that happens all the time to clear the antigens in our body. Our phagocytes or macrophages and dendritic cells are eating up things all the time. They don't go into ADEs all the time. M. Gregory says, fascinating. Absolutely. I love it. So, John, no. And John, you are from Periscope. It is interesting. I've never seen my own video on Periscope. I don't even know how to go see it. Cool. So let's see one more question here. To Ramona, the Novavax COVID vaccine has the matrix M1 adjuvant. The mRNA has no adjuvant. Is this adjuvant a concern? No, it's actually it is good. Matrix M1 will be that, remember, the original SARS-CoV has a tiny protein on it, which is called matrix protein, M protein. It is just a part of the envelope protein that is present there. So if they actually make those M1s here as well, that makes this little nanoparticle even better, because now it kind of mimics the virus a little more. That decoy duck has some paint on it as well, against which we can make antibodies too and become even more effective. So that is a good thing. So Cell Games 2006, UK just reported a reinfection in 78-year-old man with the Kent strain that had the old. So the old man had the old antibodies and they didn't work. So yes, it can pass the antibodies. The question is, what was the quality of the antibodies in that person? The immune responses in advanced ages are usually lesser, number one. Number two, the new variant, that the Kent variant, is it really reinfecting everyone? If that was the case, we will be in trouble. We are seeing cases of reinfection, 1 in a million maybe, and that is fine. That is happening. It is not happening at a level that, let's say, I was infected before and now there is a new variant and I got infected again and everybody is getting infected again and pandemic has restarted. That is not happening. Super chat. Thank you very much, Kirk. What are your thoughts about daily antigen testing to let the individual know if they are transmissible? So I would love it if we can do it, that we can have at home. Remember, in the beginning of the pandemic, I used to say a lot that there has to be a way that we can test ourselves at home. Unfortunately, there are some tests that have been approved, but even then, at least in the US, you cannot use them without a prescription, because I think our government just wants to have a very good control over who is trying to figure out if they are sick or not. For example, if I got a vaccine, I'm in a trial, and they gave me water instead of the vaccine, and now I go and get the test done, I can actually know if I got vaccinated or not. So I think they're just using us mostly for experiments. I'm being facetious here. I don't like that they have controlled these things. So 22 Dolos 22 question: it takes a few days to get an appointment for a COVID test in my area. If I start taking ivermectin, will it affect the result of the test if I get the test after I have started taking it? So the ivermectin's role is to reduce the viral load by not killing the virus, but by making the cells stronger. So in the presence of ivermectin, one can still have low level of viral replication, but our immune system would get a chance to control it fast. So you can still show up positive on the RT-PCR. So 10AE812, are you concerned that E484 mutation has 10x less affinity for convalescent plasma antibody? Interestingly, convalescent plasma was already not working very well. We know that convalescent plasma, we thought that would become the best thing, but many people's antibodies were not really working for others. The thing about these antibodies that are commercially manufactured or vaccines is that they continue to look at these thousands of strains and see what is the area of the least mutation, what is the most constant part, and they make antibodies against that. So that is the benefit. So for example, if we are the researchers and we want to make an antibody, let's say monoclonal antibody, and we want a project or startup to succeed, and we want to make billions of dollars, and we don't want it to fail, what is going to happen? We're going to put all the strains in front of us, and we are going to say, okay, guys, let's look at the genetic material here and maybe feed it into a computer and see what are the areas of least change. Then make the antibodies against those areas. That is how these things are produced. Convalescent plasmas have not been very successful because of all the tiny changes that continue to happen to genome. Cool. So this is the discussion. There's one more question here. Melanie Goodman question: would this be more effective if it was given intranasally instead of IM? So in my opinion, it could be given intranasally as well, because it just needs to be present, and there are spike proteins and our immune system are going to respond. So it could be intranasally as well. So 10AE812, excellent, getting vaccine Friday, very good, congratulations. Thrill 0415, what is the earliest that Novavax will be available in the US? So if I look at their site, this is the part that I had not done much research on yet. This is the, where is their phase 3? Here. So Novavax phase 3 has started. Study placebo, age 18 and over, check your eligibility, screening, follow-up, 2 years, randomly assigned. So what I don't know is study vaccine being researched. Can I get, cannot get, in fact, why is diversity get paid? Does the study vaccine contain, what is the placebo? Can I check to see if I'm eligible? Does a typical clinical trial? Okay. So I have to do that research to see when would they actually finish this trial. So Thrill, give me some time to look into that. I was just so enamored by their technology that I wanted to at least discuss that today. Cool. So guys, thank you very much for today's time. There is one more question. Question is by Maash. Question: I would like to know what is the point of taking any new vaccine when we have proven drugs like ivermectin, hydroxychloroquine, herbs like black seed that proven protect and cure COVID. I have discussed this before as well. If I am given ivermectin and I'm given a vaccine, I would take the vaccine too. The reason for that is very simple. The vaccine is going to prepare my immune system against SARS-CoV-2. Ivermectin is going to get my cells ready to protect against these viruses. So that is good. It is a good thing, and it has been very effective. But vaccine is just going to give me such a targeted and specific response to the virus that I know I can handle it. I know I can combat with it. So the other thing is, we should all be allowed at this time as a bridge from now till vaccine to be able to use these prophylaxis, ivermectin or hydroxychloroquine and zincs and those things. That is just incompetence, I believe. And it is, to me, it seems more and more criminal because people are dying. So to your question, answer is yes, but I am still also a proponent of the vaccine. And at the same time, I'm so sick of the leadership not to allow common folks to use these things. You channel says, Dr. Bean, you made layman understand medicine. Thank you very much. My point when I started this was to actually discuss these things with all of us and not to come here in front of you and tell you how much I know or I know medicine. I'm a doctor, I know medicine. The point was, as much information can be relayed to you so you can make your own decisions. And that was the basic belief. That was my dream to do. I thought that was the only way for us to protect ourselves better if we are all educated. Okay, so Barbara says, so it may be best to take a vaccine and then use a black seed IVM, get mild infection so we can rid it fast. I agree with Dr. Bean. Outrage about the politics of it. Agreed. Meaning these are not and or. They can actually both be done together. So this is the discussion for today. Thank you very much. There is a please like, subscribe, and share. There is a link to buy me coffees, which is in the description. There is also another link to support this channel, this work, if you like it. So please like, subscribe, and share, and you can support it as well. So thank you very much for today. I would see you tomorrow. Bye bye.